From Microbe TV, this is a special immune booster recorded during the American Association of Immunologists conference in Chicago, May 2024. Starring Cindy Leifer and Brianne Barker, and featuring Michal Tal from MIT.
Welcome.
Thank you. I'm excited to be here.
Yeah, thanks for joining us.
So we would love to know sort of how you got interested in science in the first place, and sort of how you started your career into science.
So I've given this a lot of thought over the years because people have asked me why I'm so obsessed with infectious disease. And, I've put a little bit of this out on Twitter, but my journey here was actually a very sad story of my uncle died from HIV and the AIDS that developed from that. And then my cousin died of meningococcal meningitis when I was in high school. And from the time my uncle had died when I was 8, the thing is at the time he didn't even tell us he was sick. There was a lot going on culturally. This is 1988 at the time. There was a lot going on in the world, and he was a gay man living in San Francisco. And he was afraid to tell us, I guess, so even though he was sick for a very long time, and this was, not a surprise coming, it was a surprise to me. And for me, it came out of nowhere. And that was my first realization that a bug so small you can't even see it could kill you. And that just blew my mind, and I couldn't stop thinking about that. And I thought about that for many years and tried to understand it. And then I was a senior in high school when my cousin died, and he had a date on a Friday night. He canceled it because he wasn't feeling well. Monday morning, my aunt told him to drive straight, to get an ambulance straight to the hospital because he had really deteriorated over the course of the weekend. He got to the hospital, and before they even figured out what it was, he'd only been at the hospital about an hour and he passed away.
Oh my gosh.
Wow. So, there was, people always think, if you get sick, you'll go to the hospital and they'll fix you. But I think people don't realize with some of these infectious diseases, like if you're not vaccinated, there may not be any time. There's no, with something like meningitis, there's often not time. So fast. And my aunt and uncle later learned that there was a vaccine for meningococcal meningitis that at the time it wasn't on the schedule. They actually worked really, really hard to get to get legislature through in Connecticut to get that on the schedule. Since then, there's been improvements in the vaccine and it made it into the pediatric schedule even. But, I remember my mom took me straight away to get vaccinated but how unbelievably crushing to find out that, you've lost somebody to a preventable disease.
That's terrible.
Yeah. And that really, so from there I trained to be an EMT, and then I did undergrad in biology, and I discovered immunology, and I was just, I took a microbiology class. I was into it, but I took an immunology class and I was just like, this is what I want to do for the rest of my life. The answers are here.
Very cool. So then did you go straight into graduate school after undergrad?
After undergrad, I went to graduate school. I got to Akiko Iwasaki, who was my PhD mentor. And that was actually really important to me because I really wanted to study immunology. And at the time, a lot of the really famous women in immunology hadn't had children. And I knew I wanted to have kids. That was something that was really important to me. And when I met Akiko, she was just, I saw that there was another way. And I think a lot of people see that with her now. She really emphasizes, creating a safe space for women and for women to have kids and to, so I did my PhD with her, and then I did my postdoc with Irv Weissman at Stanford. And I actually had so much fun during my postdoc. I stayed and stayed. My postdoc timed out. At Stanford, you're supposed to time out at 6 years. And I was like, I only started asking the right questions year 4. I was like, I can't go. I'm not ready to go. And Irv actually brought me on as an instructor in the institute. And I got to stay an additional 3 years after that as an instructor at Stanford. And then got this position at MIT. So
very cool. So what types of things did you work on in Akiko's lab?
In Akiko's lab, I was working on physiological processes that impact our response to viral infection. And it's so funny because at the time I thought I was doing this really basic science that wasn't going to be super relevant for a lot of human disease, and then at the end, I was working on flu and doing a lot, more. It was really important to me to do stuff that was more translational and very human disease focused. But then, fast forward to the pandemic, it ended up turning out that a lot of my PhD work was actually super relevant and ended up shaping my thinking, a lot. So
awesome. Yeah. And then how did that transition into your time as a postdoc, and how did you make that decision?
So, grad school was going great and things were really good. And for those, who know Akiko, she's a very hands-on mentor. And I think I had something to prove to myself. Like I think I was asking myself if part of my success was just Akiko, or if it was me. And I kind of had something to prove to myself. And I had my heart set on getting into Irv Weissman's lab. Like I asked Richard Flavell, was the chair of the department at Yale, to send Irv a message because Irv wouldn't respond to emails. And I knew Irv had this big lab and people had a ton of freedom and a lot of PIs came out of there. And I think, I do think I had something to prove to myself that even just with a lot of freedom to flounder, that I could still succeed in that environment as well. And
what was it about Irv's lab that drew you there? So what did you want to work on?
He was doing so much human translational immunology and doing a lot of really kind of applied immunology, almost experiment, where that I was really drawn to that. And it's funny because when I was trying to get the position at MIT's like, why would an immunologist come to the School of Engineering?
Yeah.
And I'm like, no, I do applied immunology. So I'm an immunoengineer, but this is really, that's, the thing about Irv's lab that really kind of pulled me there was all this applied immunology. I thought that was so cool and a really cool way to investigate the immune system and all these, he was really pushing forward these concepts of new innate immune checkpoints, and looking at that in cancer, but I really wanted to look at that in the context of infectious disease.
So what was your project there? So what were you working on? So I guess you said it took you 4 years to figure out the right question. So sort of like, what were you kind of working on?
My main postdoc project was looking at this CD47 SERP alpha axis. So it's an innate immune checkpoint where, your cells have these don't eat me signals. And macrophages are coming in and they're like, what should I eat? And in an inflammatory environment where macrophages get really eaty, cells are using this to protect themselves, to be like, don't eat me. I'm supposed to be here. And this is, I found that this actually gets upregulated in the context of an infection. And part of this is, your immune system can kill you. And we have all these breaks in place to protect you from that. And it can minimize your ability to overcome this infection, but it can also keep you alive.
Yeah.
And, one of the things that came out of, so eventually out of this work, a drug came out of that, magrolimab. Irv spun out a company, 47 Inc. It was sold to Gilead. Magrolimab has been, the trials ended up stopping for increased death from infection. So the thing is, I think with these checkpoints, there's still so much we have to understand about, we're always saying there's so much overlap between cancer and infection. And I think we think it's all the same. But the thing is, cancer is more like the kind of inflammation we have in a chronic infection and an acute infection. Yeah. It's a different ballgame. These checkpoints taking the brakes off can be really dangerous. So, I think there's so much for us to still understand. I think these checkpoints are important, and I think we just have to understand better how they're impacting our responses in acute infection and how to navigate around that. But I think there's so much potential there. And then I ended up finding that there was a bacteria that was actually mimicking our own CU47 and hijacking this, turning on those brakes on the macrophages. And but that's when I started working on Lyme disease.
Yeah, which bacteria?
That bacteria was Borrelia burgdorferi, which causes Lyme disease. And so that's when I started reading about Lyme and reading about chronic Lyme, which a lot of people, there's this whole thing. Is chronic Lyme real? What is this? And is the bug and as I started reading about this, one thing that really just overwhelmed me was I was reading this review, and they were basically talking about how chronic Lyme isn't real. And it was a description to clinicians about how they would be doing a disservice to their patients by even entertaining this, because this is psychiatric, and this is, and part of the rationale there is this female to male skew, that it's 70-30 female to male. And that's part of this justification that chronic Lyme isn't real. And I was just, I read that, and I was just like, you know what? I need to be here in this space. That's when I started asking the right questions, I think.
Okay.
Because that's when I started asking, what is it about the female immune response that is predisposing to infection-associated chronic illness? And then, there was a mouse that changed my career. So we're running these Lyme infections, and I became I became a complete feminist in my science. I wanted to compare everything females to males. I wanted to do more stuff in females than males. I really wanted to start to understand the female immune response to infection. And I wanted to understand also what was going on in the uterus. I saw that with Lyme, everybody looks at all these organs, and everybody's looking at the bladder, and nobody's been looking at the uterus. I mean, there's been one paper looking at Lyme in the uterus. And so we started looking at uteruses. And one day we go down to, collect uteruses from these Lyme-infected mice. And I had some of the people in my lab were down there, and they'd gone down with a little 24-well plate to just pull out these little uteruses. And there was a uterus so big, it needed a 50 ml Falcon tube. And they called me over, and they're showing this to me, and I'm just like, oh my God. I mean, this mouse was in so much pain, and this is so horrific. And it was chock full of Borrelia. And it was just like, this mouse completely changed my career. That's how I ended up at the Center for Pathology Research. I had a meeting at some point with Linda Griffith, who's the director, and I was like, do you want to see something gross? And she's like, of course.
Because that's what we do.
Yeah, exactly. Because who doesn't, right? And I show her this enormous uterus that's, so pathological. And she's like, we got to get you over here. And that started, all of this, bringing all this work about asking about some of the connections between bacterial infections and gynepathologies. And, there's a lot of exciting stuff going on.
So I guess in general, what types of things do we think about in sex differences, or what do we know about sex differences between men and women?
Yeah. So there's some
immune responses. I don't mean like in general.
Yeah. There's really incredible work that's been done. Like some of the work that's really inspired me has been some of the work by Sabra Klein. And, so much work looking at acute infections like flu infection and showing, age-matched male to female, males can have a much more severe, even death response to these acute infections than females. But most often, they'll survive and then do great, right? Whereas females have a milder acute infection, but then are more likely to go on to chronic illness. So, I got, a lot is being asked right now in this acute infection space. And where I'm really trying to focus my efforts is trying to understand this chronic, this tendency towards chronic illness, and why is that? And what's different there about, what's different from those initial responses. And part of it, I think, is, this whole thing with the man flu, right? And I think it's so funny that we get this name as if we're the complainers. This whole reporting bias that women are more likely to report, adverse events to vaccination, all that, as if that's a reporting bias and not a biological. But then when you look at the actual response to infection, you've got your man flu, right? So you've got the guy who's like, I can't move. I can't get out of bed. Like, somebody feed me. And you've often got this woman in the picture juggling 1,000 balls. She's not feeling great. She's not debilitated. She's still doing all this stuff. And I actually think we have a ton to learn from men about resting aggressively. I think we shouldn't laugh about the man flu. I think we should learn from it, because I think that's how you don't end up with chronic illness later. I think you take the time to give your immune system the space to divert energy over to the immune response, to handle it, to resolve it, to overcome. And I think if you are just kind of keeping it at bay and doing all these other things, I think it comes back to bite you sometimes, so.
That's a really interesting hypothesis. Do you think that the stress also plays a role in that?
I'm sure it does.
Yeah.
I mean, we know how much stress impacts immune responses, and we know that you can keep something at bay for a time. Like, if you need to,
Right.
You can use adrenaline for all sorts of things. You can be like, I have to do this thing, and then I'll let myself fall apart.
Yeah. Yeah.
But the question is, what does that cost you?
What is the cost? Yeah. To your health?
Yeah, that's a great question. So what other types of things have you observed in terms of sort of the kind of pathological effects with infection that maybe we wouldn't have noticed? I would never have expected to hear about changes in the uterus with Borrelia infection. Yeah. So are there other types of changes like that you've seen, other types of pathology?
Yes. So, I feel like this is the space I'm most excited about, and it's all totally unpublished. Hopefully there's somebody in my lab working on this uterus paper right now, Paige Hansen. So she, hopefully she'll get this out soon, but there's all this other stuff that we're seeing that's different between the males and the females, in the animals, in people who are participating in our clinical study. So in terms of how these chronic illnesses manifest, I think there's not just differences between males and females in terms of risk of developing the chronic illness. I think also how these chronic illnesses, the symptoms that are most common in males versus females, we're also seeing important differences there that really align with what people are reporting in long COVID. And I think there's just so many profound similarities between the infection-associated chronic illnesses, almost regardless of the infection that took you there, because you've got this bacteria and you've got this virus in there, but at the end, this looks the same. And so there's a lot of questions there about why is that? And, so that's where we're diving into that, and why does that look so different?
Okay.
Males and females.
Is there a lot of overlap, do you think, with autoimmune diseases as well? Because we know that's another area in which there's a significant female bias.
Huge.
Yeah.
And for sure. And some of these, are comorbid with autoimmune. And I think there's also a ton that I think we've bucketed as autoimmune that I think we oversimplified, that I think is more complicated
Might
than
be
that.
these post-acute syndromes.
Well,
Infection?
so more like when we talk about a classic autoimmune, we're talking about a break intolerance. And you're now actively attacking your own self-proteins.
Yep.
But let's imagine the scenario that you have now mounted pathological responses against some microbes. Let's say against conserved features in microbes that show up in other microbes. So let's say, what if you now are mounting a highly pathological response against your cell flora? Well, what would that be called? Is that autoimmune? Is that a loss of tolerance to your ecosystem that lives with you? Is that what is that?
We need a new name.
Yes.
And it's more complicated than just autoimmune, because there's no break intolerance there, but it could still be really bad, because if you're now constantly getting sensitized or constantly attacking organisms that live in you
and do important things
for you, then what's that going to look like? And what's that going to mean? And what would that be called?
Symbioimmune, autoimmune, symbioimmune. I don't know. Yeah. It's very interesting.
We need a name for it.
We do.
Yeah.
You have to think about that and then write a paper on it.
So you call yourself an immunoengineer.
Yes.
Is that mostly because you're working on sort of applied immunology, or are there other pieces to that term?
Well, so I, during my postdoc, I really wanted to map out as much as I could about the immune responses to Borrelia and how they were changing under different scenarios. And so I started looking at how could we ask the immune system more open-ended questions? And I think this is really important, because I think currently we ask the immune system very pointed questions that then actually give us very low resolution information back. So for example, with the Borrelia tests, because we've really struggled getting a good direct test, we do this indirect testing where we're looking for antibodies against the Borrelia to see if you've had a Borrelia exposure. But then that's all it tells us. It just tells us we're looking for IgG against certain Borrelia epitopes. And so the only information we get back is, have you had an exposure? It doesn't tell us if you're still infected. It doesn't tell us how that infection went. And I started asking, well, what if we use the Borrelia to probe the immune response to the Borrelia and just ask an open-ended question? Just ask the immune system, how are you responding? How is that changing over time? How is, using this as a bait to pull out all of the Borrelia specific antibodies and look at all of it and kind of, so we call this the flip, partly because it's, flow-based immune profiling, but partly because we're flipping how we're looking at the antibody. So instead of looking at the variable region, looking at the FC. Because I really think the FC is the business end, where this isn't just a biomarker. This is an immune effector protein, and the flavor of inflammation that it's going to trigger is all there in the FC.
Yeah.
So while everybody's been so hyper focused on all these, epitope mapping and like antigen specific responses, I'm like, it binds Borrelia or it doesn't bind Borrelia. But I honestly don't care about the epitope. I just care about the FC. And I think that asking the immune system about FCs is a way to ask a much more open-ended question about, how is this response going? Is this a protective response? And this person's going to recover, never think about Lyme again. This is a nothing. Or is this a not protective response? Is this a catastrophic response that's going to bring the house down? And those look really different. So I think instead of just asking about exposure, we could be asking about how things are going in a way that's much more open-ended. And that's what I'm super excited about. And I think of this as immunoengineering. I think I'm, engineering tech to allow us to ask open-ended questions of the immune system, and ideally eventually try to engineer those responses to, because I think people have looked at this as a paradigm of like, do you need antibiotics or do you need steroids? And I'm like, anti-inflammatory versus inflammatory is this dichotomy that this is not, inflammatory means 1,000 things.
Yes.
And so I want us to be thinking about flavors of inflammation and how you nudge towards a protective response. If you can define what's a protective response and you see that a not protective response is happening, how do you nudge it in the right direction? And that's where I think a lot of that immunoengineering comes in.
Very cool.
I'm excited about it.
So I wanted to touch back just briefly before we close out. You mentioned that you've had some really great mentors along the way. Can you talk a little bit about some things that you've taken from what you've experienced and how you think about mentoring the next generation of scientists in your lab?
So one of the greatest pieces of advice that I've gotten is to work with nice people and people that you like. And I think there's so much of the job is what we make it. And we kind of pick and choose which parts of the job we're going to do and which parts of the job we're going to, if you email me, I probably won't respond. I've given up on responding to emails. But, I feel like people who carve out, like, what are the features, like making it the job they love to do. So many of the things about the job are hard. But if you make the job your dream job, like I feel like, and you get to work with people you really enjoy working with. And like, what an amazing, some of the other, like constantly being broke and begging for money and like always running out of money, that sucks. But getting to spend every day working with people you really respect and enjoy getting to, have this back and forth with, I think that's amazing. And I think for me, the job is really so much about the people you work with and the people you get the honor to train,
That's fantastic. I think that there's so much more to learn about so many of these topics that we could have this conversation go on for a very long time. But unfortunately, we're running short on time right now. Thank you so much.
Thank you. Thank you for having me.
That's a special episode of Immune and Immune Booster. If you have questions or comments, please send them to immune@microbe.tv. If you enjoy our work, please consider supporting the science shows of Microbe TV. Go to microbe.tv/contribute. Microbe TV is a 501c3, so your donations are U.S. federal tax deductible. Our guest today from MIT, Michal Tal. Cindy Leifer is at Cornell University. Cindy Leifer on X. And Brianne Barker is at Drew University. BioProf Barker on Blue Sky. Music on Immune Boosters is by Tatami. Thanks for listening to Immune, the podcast that's infectious.